So these are some of our laboratory samples, and we commonly use carrots as a baiting technique for the fungus. And so this is not us, this is an accepted protocol for Ceratocystis fimbriata. It cuts down on a lot of other fungi growing, so it's a good technique to bait. We use it with wood, we've used it with soil, we've used it with insect frass, and within 1 to 2 weeks you tend to see if Ceratocystis is starting to grow. Kind of a darkened area on the carrot, like that. And so when you look under the microscope, you'll start to see fungal structures indicative of our rapid Ohia death pathogen, the Ceratocystis fimbriata. And so this is actually what the pure culture is. So as JB mentioned, we're trying to figure out the overall distribution on island. And some of the biological studies is trying to figure out where it came from, what did it come on. So we're trying to establish a population. So right now we have close to 100 different isolates collected around the island to see, are they genetically the same thing? And that's the type of study, and this is, so we end up with pure cultures of the fungus. So can kind of go to the back wall to show you an idea. This is our wall of samples. So as JB and Flint mentioned, they're usually the ones getting calls to go collect samples usually. And so they'll bring back portions of wood, cookies, sometimes sawdust. Again, we've looked at soil, and we apply the carrots actually directly to the wood pieces to help bait the fungus. So the fungus is well established usually in the wood. It's key to see the internal discoloration. So JB mentioned Ohia die potentially for other reasons, but internal discoloration, that discolored area usually has a ring or a starburst pattern, and especially if you take kind of the bark off and you can see dark stained lines, that is essentially the fungus. So we can place carrots in those areas and the fungus starts growing on those, and then at the right stage, usually a week to 2 after being together, we can then isolate the organism off of the carrot and start purifying it on the plates. So this is what it looks like when it's growing as a pure culture. And these are the kind of the light peach, orange, sticky spores that you heard about. And so what you're looking at is actually the mycelium of the fungus, which then produces sexual fruiting bodies called ascomata. And that is the house for sexual spores called ascospores. So you can see what looks like maybe a flask-shaped, round base with a long neck, and this sticky, actually somewhat fruity smelling spores are the orange portion of the plate. Yeah. So yes, you have to have the susceptible host, the Ohia, you have to have the fungus, and then the critical environmental components. So that's the type of work we do here, really understanding pathogenicity, how we proved pathogenicity the first time, going through and completing what's called Koch's postulates, being able to take a healthy seedling and a pure culture and the right environmental conditions, inoculating it, and actually recreating the symptoms you see in the field. Well, it's definitely what we're trying to do with this population genetics study, but I guess as a rough time frame, the dieback of the 80s, early 90s pathologists were working on that project, looking to see if there was some kind of biological association, meaning a fungus or Ceratocystis fimbriata was not found at that time, was not found associated with Ohia at that time. So if I had to guess, that's kind of a time frame of from that point forward. A lot of times tree diseases caused by this fungus could take 2 to 3 years to actually get the symptoms in the field for one tree. So the public really became aware and started contacting JB and Flint, you know, that time. So it's, you know, it's really trying to back up to get a better understanding. When did the first signs of it occur?